Good morning, everyone. I want to thank the organizers for giving me the opportunity to speak today. My disclosures are here related to research grants. And the goal of my talk today is to present approaches that may assist in defining microbiome interactions with a disease or condition. Since we've worked predominantly on colon cancer, I'm going to start there, and then at the end just show you a little bit of data from HIV. So my laboratory works in 3 areas. I'm going to focus on the top 2 today. In one case, it's how individual organisms can contribute to a disease process, and the second is how communities of organisms may contribute to disease. All the work I'm going to show is translational, involving both human samples and preclinical murine models. And I think it's safe to say, and you would all agree, that microbiome work is interdisciplinary, a true example of team science requiring a multiplicity of approaches. And with that in mind, I use this slide to illustrate why I don't think we're going to be sequencing our way to translation, to bring something to the patient's bedside. And I use an example these 2 outstanding papers in Nature Medicine that have many other aspects that are important. They looked at over 900 fecal metagenomes, or shotgun sequencing between them. And the key point is that in both papers, when they look carefully at the predictive power of the data for colon cancer, it fell just over about 80%. And adenomas were either not included or had a much lower predictive signature. And the point is that you can't miss cancer. So if someone had a positive test, as discussed here, they needed a colonoscopy. And if they have a negative test, they need a colonoscopy. So in this case, this predictive value fell short of what is needed to take care of patients. The reason we study colon cancer is twofold. One, we are in the midst of an emerging epidemic of early onset colorectal cancer in individuals under the age of 50, dating back really as far as 1970s cohort. This is a feature of health disparities, and shown here are 2 individuals, both with stage 3 colon cancer, and as we know, Chadwick Boseman lost his life recently to this disease. This is a global problem. So it's, in my view, an important problem across the globe, even in areas of the world such as Africa, where we expect colon cancer to be less frequent. So the overarching goal of our work is to try to corral the microbiome to inform prevention and therapy in human colon cancer. So this slide illustrates the idea of single microbes can be important. I want to thank Dr. Xiaoguan Wu and Franck Cousseau for their work, and I'm going to focus on a bug we've long studied called enterotoxigenic Bacteroides fragilis. Humans are commonly colonized with both Bacteroides fragilis and the strains that make a toxin. And what we've learned over time is that this toxin is key to pathogenesis. If you take that gene out of the genome of the bacterium, all the tumor effects and signaling effects go away. We understand the toxin can induce DNA damage. It triggers pathways in the host that are linked to colon carcinogenesis. And in fact, this can happen very quickly in a mouse within a week of colonization. Of course, then the tumors grow. Now, this isn't the only example in colon cancer. There are other organisms, PKS E. coli and Fusobacterium, they're important, but it's an example of how one bug can teach you a lot about a disease process. Now I'm going to talk a little bit about community as a driver of colon cancer pathogenesis. And here I'm going to talk about biofilms, which are invasive aggregates of bacteria that directly interact with the colonic epithelium and are common in colon cancer. So by that, this cartoon illustrates what I mean. The entire colon is covered with a dense mucus layer and a loosely organized mucus layer. And that loosely organized mucus layer can be invaded by bacteria is shown on the normal side of this, but a biofilm forms when that dense inner mucus layer, which is normally nearly sterile, is invaded by bacteria. So the pictures on the right illustrate binding of fluorescently labeled probes that illustrate these bacterial populations. So in the healthy colon here, you can see the mucus layer in light blue. But when it's invaded here by a polymicrobial biofilm and here by a biofilm that's predominantly in green, Bacteroidetes, and in yellow, Fusobacterium, these are striking structures on the mucosa. We've been able to show that these come sort of in three flavors. The majority are polymicrobial, and then there's a large subset that actually have invasion with Fusobacterium, and a much smaller subset that appear to be proteobacteria, gram-negative rod dominant. And when you look at colon tumors, you can see these are particularly prevalent on the right side of the colon in both US and Malaysia cohorts. And in Malaysia in particular, these are common in the left colon for reasons we don't fully understand. However, when you look at individuals undergoing screening colonoscopy, about 15%, whether on the right or the left colon, display these biofilms. Now, does it matter? Does this change the function of the tissue? And I don't have time to show all the data, but the red starred areas are steps that we've been able to identify. So I just showed you biofilm formation. And when we look carefully, and here's a picture of this, using immunofluorescence staining, we can see that there's a change, a likely change in barrier function because there's decreased E-cadherin, a critical intercellular protein in the colon. So here on the left is the biofilm positive sample, and on the right is the biofilm negative. On the top are colon cancer patients, and on the bottom are screening colonoscopy patients. And you can see in the normal condition, whether in colon cancer or screening colonoscopy, there's a lot of E-cadherin staining, and this is markedly diminished in the setting of a biofilm, strongly suggesting a decrease in barrier function. In association with this, we see immune activation, we see phosphorylation of STAT3, and we see the precursor to tumors, colonic epithelial cell proliferation. This is shown here. Again, these are colonoscopy biopsies, and this is IHC, Ki67, which is a marker for cell proliferation. And you can see there are fewer brown cells in the biofilm negative sample than in the biofilm positive. And all these results can be quantitated and show the same results. We were also working with Gary Zustak's lab. We were able to show that there's a unique polyamine metabolite that is associated with biofilms, and this is N1N12 diacetylspermine. However, this doesn't tell you there's carcinogenesis. So really the next question was, are these biofilms carcinogenic? And to answer this question, we teamed up with Christian Jobin's lab at the University of Florida and work done by Sarah Tomkovich and Christine De Gea. And we looked at a slurry made from biofilm positive patients that we then put in germ-free mice that are susceptible to colon tumors. And what we were able to identify is that all the biofilm positive samples, which here are the tumors, biopsies, or the normal flanking from the tumor patient, led to induction of tumors in the colons of the mice, but that were not seen when the mice were inoculated with biofilm negative biopsies. Dr. Julia Drews in the lab has taken this a step further, now taking the examination of this to individual patient samples as shown here. And what we've learned is not every biofilm will induce tumors in the colon of a mice. However, some do, whether they are biofilm positive or biofilm negative, as shown in this slide. In green is the biofilm negative, and blue is the biofilm positive, and here is the control, sham, as well as here is the control of the slurry that was initially examined. She has now been able to culture bacterial isolates out of this particular sample, 3728T, And using these 30 isolates as shown in this graph, she can actually recapitulate the tumorigenesis from the slurry. So this gives us a microbiology result and a way to try to understand exactly which organisms and their interactions may be pro-carcinogenic in the colon. I want to mention one more thing about biofilms, and this was sort of an accidental observation. In the times we were collecting colons and examining biofilms, Christine De Gea was fortunate to get a few samples from individuals with a rare hereditary syndrome for colon cancer called familial adenomatous polyposis. And what, in this case, patients were getting a colectomy to protect them against colon cancer. And we learned a couple of things. First, the staining was very different than what we saw in sporadic colon cancer. And in fact, the majority of the bugs on the surface of the colon were either an E. coli or a Bacteroides fragilis. And second, these biofilms and red dots actually went all along the axis of the colon, suggesting that once a biofilm forms, it may be quite extensive in the colon. And they were seen on adenomas or on the normal tissue. Additional studies traced these back to the PKS positive E. coli I mentioned and the enterotoxigenic Bacteroides fragilis. And in studying those organisms' interaction more, we identified that they actually, those virulence factors interact and are synergistic in their action in tumor formation. So there's quite a bit that can be learned through microbiology, virulence factors, and different disease models. Now, microbiome research, you might say it's been dominated by big data, as I mentioned up front with the metagenomes that I mentioned. There are a variety of big types of data that can be used to address microbiome research and to try to derive both microbial and host signals. So one possibility is databases. These are large data, listed here are some large databases that have been used. Then there are sequencing methods, and this, the most commonly used still is probably 16S RNA amplicon sequencing that just detects bacteria. But metagenomics, which is shotgun sequencing, which allows you to assemble the DNA and can allow you to pick out both microbes and host responses, is another possibility. As well as bulk or single cell RNA-seq, again, can be related to either microbes or to the host, and TCR-seq if you want to drill down to exactly what's happening in the immune system. I'll also mention a couple others: computational meta-analysis, which I'm going to introduce in a moment, metabolomics, which some think are the final common denominator in microbiome research, proteomics, still in development, and ultimately machine learning and artificial intelligence, I think we will see applied. So I'm going to give you two examples here. This is the work of Jiajia Zhang, and we had the opportunity to get data from the Clinical Practice Research Data Link, which is the largest electronic health record in the world and housed in the United Kingdom. And with the help of their research team, we were able to set up a case control study matched by year of birth, gender, practice site, and the registration year. And these data stretch from 1989 to 2012. And what we were trying to look at was the relationship between oral antibiotic use and the subsequent incidence of colon and rectal cancers. And so the basic results are shown here, and there were a couple surprises. So in the colon and on the x-axis is the total exposure to oral antibiotics, and on the y-axis is the onset or the detection of colon cancers in association with that antibiotic use. And what you can see, with as little as 15 days of antibiotic exposure, there was an uptick in the detection of colon cancer. In contrast, in the rectum, there was a decrease in association with oral antibiotic use, but it was slower and it plateaued at about 60 days. When we looked at more detail at the colon, all of the signal came from the proximal colon, which is where the oral antibiotic concentration will end up being the highest in the colon, and there was no signal in the distal colon. One aspect of this data that made it, if you will, in my mind, more believable was that the antibiotic exposure association with the onset of colon cancer was more than 10 years prior to the diagnosis. So it was not near the time, which might have been a confounding effect of the onset of the cancer, but really distant from the cancer with a subsequent onset. The other method I want to mention is what we call a computational meta-analysis. And in this, a key feature is that we're reanalyzing the raw sequence data, not the published results as in a typical meta-analysis. And in our case, we use a high-resolution pipeline called Respira Insight. And the reason we use that is it has a higher species-level assignment than some other commonly used tools. So in this case, in colon cancer, we looked initially at 11 cohorts, nearly 500 colon cancer patients, and we're able to identify a consortium of 5 organisms consistently enhanced in colorectal cancer, whether looking at healthy or at normal tissues in a cancer patient. And then we were able to find identical results in our colon cancer cohorts as a validation cohort. And it's exactly that approach, working with Sue Tunham and Adeline Koa and James White, that we took to address, to take a look at how the HIV microbiome looked across studies. So the key points of this study is, this is from the first papers we found, which were in 2013 until the end of 2017. And at that time, we came across 22 studies with 17 available data sets. We again looked at 16S rRNA amplicon data. Now this represented over 1,000 samples, 300 HIV negative and about 700 HIV positive. And I also want to give great credit to all the authors of these papers who all worked with us to get the data sets right for this analysis. We were able to publish our first paper looking just at alpha diversity across these data sets in clinical infectious diseases last year. And what we learned was in the stratified analysis in which we controlled for HIV, gender, and sexual orientation, we identified a decreased alpha diversity only in women and men who have sex with women, not men who have sex with men and persons living with HIV. And the overall results suggested that HIV, gender, and sexual risk categories should impact alpha diversity and should be considered in analyses. We're in the process of taking these same data and now trying to look at the taxa associated with HIV and sexual orientation. And our preliminary results suggest that MSM status has a stronger impact on the gut microbiota than HIV status. One of the issues in across the microbiome field has been many small studies getting published with limited clinical metadata, and I think this is an area where the entire microbiome field perhaps can do better over time. I just want to mention 2 challenges in the field. One is that there can be marked technical differences between labs and between papers. And this is emphasized by the Microbiome Quality Control Project led by Curtis Huttenhauer, looking at differences amongst 15 labs. And the second thing I want to emphasize is how we probably need to think more broadly about the exposome. There appear, as we all know, many factors that impact the microbiome beyond diet and medications. Genetics and epigenetics probably matter, as well as disease interactions. So this is something in collection of metadata that we certainly need to consider. And lastly, I just want to thank everyone in the lab, our collaborators at Hopkins, our collaborators outside of Hopkins, and all of our funders, which has included CFAR for the HIV work. Thank you very much.